Hi, all. Welcome to our presentation upon genome editing. Today, we'll be talking about taking organs from pigs and transplanting them into humans and doing that through gene editing. My name is Katie Strickland.
My name is Cody McMillan.
My name is Mark Bentig.
I'm Will Sutherland.
Yes, and today we'll be talking about how money grows on trees, but organs grow in pigs. And this line we kind of came up with as a joke, but honestly correlates to just the genome editing and why this topic started getting researched.
Yeah, so a quick overview of what we're going to be talking about. So new advances in genetic engineering have made it possible to create knockout pigs, which is where you modify the genes of a pig to make its organs transplantable into humans. This is xenography, and you have to modify them because the human body doesn't typically like using pigs from other animals, so they have to be genetically modified. So why are we doing this? The big problem we're trying to fix is that in the field of organ transplantation, there is a huge discrepancy between the amount of patients who need organs and the amount of organs available. There are more than 100,000 people on the wait list for organ transplants in the US alone. And each modified pig can provide 2 kidneys and a heart. So if the scale up continues, it'll relieve a lot of the stress on the industry.
So in competition with xenotransplantation is organ procurement companies and organ donors. However, the demand largely outweighs the supply, so this is not sustainable on its own. Furthermore, vaccinations have made previously unusable organs available, such as HIV vaccinations, but as Will mentioned before, the demand is still much greater than the supply. It's also important to note here that xenotransplantation dates back to the 1900s, but it consists of mostly failed attempts. Past attempts on the human body were unsuccessful as the human body rejected the organ and initiated an immune response that resulted in either death or infection. In 2000, pigs were being cloned and genetically modified, and their organs started to be tested on non-human primates. Around 2009, CRISPR was founded, and as early as the 1950s, gene editing was starting to be explored and experimented with. So xenotransplantation is not new. However, the idea of editing these genes so that they are more compatible with humans is new, and we're starting to research that effect. Why pigs? Pigs are just mammals like us, and they have a very compatible genome. They are readily available, they exist in large numbers, and they're very easy and cost efficient to breed and raise. Furthermore, the size of the pig organs are very similar to that of humans. So as I mentioned before, gene editing and CRISPR, they're not new. However, we're starting to expand gene editing to look at how we can edit animals that are similar to humans to further benefit humans.
So how are we modifying these pigs to make them knockout pigs? So when the pig organs are transplanted into humans, they're quickly rejected by like a strong immune response called hyperacute rejection. And what causes this is an antigen called gal, which is found on the surface of pig cells. And there's a specific enzyme called GGTA1 that produces this antigen. So our solution to this is to inactivate the gene that encodes the GGTA1 enzyme, which itself produces the gal antigen. So if we can get rid of the enzyme, we get rid of gal, we can put the organs into people.
So again, the main component here is gene editing. So we want to use specific nucleases to help create changes in the DNA sequence. The main function of this is to break the DNA double strand and then trigger the cell's repair mechanisms. These mechanisms allow for the creation of the knockout animals or the genetically modified pigs and allow a more efficient gene editing process than previous methods. Specifically, we're using ZFN, a zinc finger method that replaces the DNA binding domains with different bacteria. So first, the pigs have to be cloned before they are used with humans, and DNA integration may occur randomly here. There are point mutations, insertions, and deletions used with the modern gene editing, and as equipment progresses, this gets to be more specific and accurate. As mentioned before, the DNA binding with the zinc finger motif, or the ZFN, that's when we're recognizing the specific nucleotides we want to change, delete, insert, mutate, etc. The ZFN binding domains are then replaced with proteins and bacteria, and this creates a positive feedback loop where we can keep editing the genome of the pigs. Like I said before, this project is still in early development. However, there is a strong foundation in gene editing with human genomes, and now we are simply moving on to the pig genomes and how we can further incorporate that into the human body.
Yes, continuing what Cody said about how to incorporate this into the human body, this figure is a wonderful example of that. It's correlating the human serum antibody binding to the pig red blood cells. And so it's pretty much saying how compatible they are and how well they're staying compatible throughout their interaction. So the top line, the first two, is the antibodies of human blood cells and then a pig that is not a wild pig, so a pig that is not genetically edited. And as you can see, there's a lot of different variances and they're very different and they don't interact very well. However, the bottom line, or the B's, are whenever they do interact very well, and it's the edited pig. And so the edited pig is able to interact with the human body much better, to where the human body isn't attacking that new introduction. And so how would all of this connect to the course? We use signal activation and transduction in this gene editing. And so editing these pig genes to insert these organs into humans, they use the ADP and the ATP to inhibit cellular process in pigs that our bodies don't like, such as adiposis. And so using the concept of inhibitory constants, as we talked about in class, was a big factor in how the scientists tackled editing these genes. They also utilize the activation pathways that complemented each other, and we've been talking about this in class with positive feedback loops and how those activation pathways enhance and affect other activation pathways, and also to avoid the chronic rejection of these organs in the human body. This RNA was modified to interfere with this process. This RNA modification, like we talked about, limited infection, and this was something we talked a lot about RNA and gene expression in class. In fact, this is a picture of a process of how the gene was expressed in class. And so this is a very simplified version, but it's what they used in this gene editing process of how to modify the RNA so it was expressed properly in the human body once it was transplanted. And to continue about how this relates to engineering principles, so this gene editing controlling gene expression is very much of an engineering principle of learning how to optimize a process and make it the most efficient and the most last the longest. And so editing these genes makes this organ have the longest lifeline in the human. You're also controlling this process in the human beings. So these scientists are having the chance to completely change people's lives and change the world for good, which is a principle in engineering that we at UT also get a big emphasis on. And then finally, these scientists and using these pig organs and just organs in general are treated as a unit in the same way that the cell is a reactor. This pump, or this heart, is considered a pump. And so you can kind of view the pump as one unit in like a grander scheme of unit. So like connecting to a chemical process flow sheet down below, this pump is the heart, and it's connected to all of these other body parts in this functional system.
So other relevant market opportunities for the same type of technology, which would be gene editing and gene engineering, would be what a specific group of scientists are trying to do is resurrect the woolly mammoth using material extracted from fossils and remains and well-preserved mammoths, and adapting it using DNA of similar species, such as the Asian elephant. And so that's just another different application, but the same technology and a very similar gene editing knowledge that's used. So as with any new technology, there are always ethical concerns that go along with it. In this particular case, one of the primary ethical concerns is the breeding and generation of pigs for the express purpose of killing them to harvest their organs. One could argue that these are going to save human lives, which are more important than pigs. Regardless, it presents an ethical concern that needs to be thought about before proceeding in such large numbers in a scaled up fashion. Secondly, there was a New York Times article that stated that these early trials were done on brain dead patients, which presents another ethical concern. Though they are unaware of what's going on, did they relinquish the autonomy over their body for those experiments to be run on them? The NIH also has stated that non-human organ donors may harbor infectious agents, particularly viruses. And so this would be another concern, practical concern, but also an ethical concern that we need to think through before transplanting non-human organs into humans. We wouldn't want the human patient to get sick or mutate a virus and start to spread that to a lot of other people. And to quote from the Jurassic Park movies, Dr. Ian Malcolm, your scientists were so preoccupied with whether or not they could, they did not stop to think if they should. And so here's a table that expresses the survival time for primates where xenotransplantation was done. You can see that there is a long survival time in some cases for heart transplants, but many more primates died within a month because of the xenotransplantation, whether it going wrong or the primate's body not accepting the organ. Again, another thing to think about when discussing the ethical concerns. In conclusion, the domestic pig is a viable option for this application in the sense that it is anatomically and physiologically similar to humans, and pig breeding is efficient and cheap. Some of the weaknesses is that genetic engineering is not perfect. It's very hard. There are unintended modifications that occur. We've talked about this in class. And specifically, the long-term effects of transplants have not been studied, so we don't know what happens 10 to 15 years from now. We'll only figure that out 10 to 15 years from now by studying the patients. And so ongoing research is needed in this, and to limit the rejection of the organ in the human, to continue ensuring that the human body accepts this foreign substance, if you may. And so more research is needed there. And there are several applications for prospects for the future in terms of many other fields of biology and engineering. So thank you for listening to our presentation, and our references are as follows.